Yeah, good afternoon to all of you. I'm very happy to formally introduce Professor Sreelaja Nair. I and Sreelaja are colleagues in the Department of Bioscience, Bioengineering. Sreelaja primarily, she's actually a biologist who asks questions primarily at the cell and molecular level, and she's specifically a developmental biologist, and she is trying to understand mechanisms that lead to how a zygote that is formed by diffusion of a sperm and egg develop into a full-fledged multicellular organism. This year's Nobel Prize is for the discovery of microRNA, and it turns out that microRNA are very much involved in this developmental process. So in our department, in fact in our institute, she is the most appropriate person to tell us about this year's Nobel Prize. And before I hand it over to her, I just want to share with you a cartoon that appeared in Times of India in October 23. There are two children who are selling lemonade. There's one child who has written lemonade and 50 cents a glass. Nobody is there. And right next to that, another kid. He's selling AI lemonade and $20 a glass. It's a big line. I'm reminded of that cartoon because I have never seen this many people in a lecture on Nobel Prize event all these years. We started in 2009. And I think it's making sense. Fortunately, I have that cartoon still with me in my mobile. Okay, Sreelaja.
I'm here to talk today about the 2024 Physiology or Medicine Nobel, which was awarded to Victor Ambros and Gary Ruvkun for their discovery, as the Nobel citation says, for their discovery of microRNA and its role in post-transcriptional gene regulation. So these days I've been teaching BB 101. So I decided to pitch it at that level and maybe slightly higher than that. So I hope I'm able to relate to all of you, and I see a lot of students in the audience, so maybe it was an appropriate pitch. Anyway, you will know in a few minutes. So before I get to what both these scientists did, I want to give a short introduction of the context in which this discovery was made. So all of us are made of many cells, and all of these cells have the same information in the cell's nucleus, which is the DNA, right? So as human beings, we have 46 chromosomes, 23 pairs, 2 of which are 22 autosomes and 2 sex chromosomes. And within these chromosomes, there are linear segments of information which are known as genes. These genes get made into a different version of this information known as RNA. This RNA then gets translated into a structure, a three-dimensional structure called a protein. And all the different cell types in our body or in any body in a multicellular animal arise because in each cell, different sets of these proteins are expressed. So there are 4 examples shown. This is a neuron. This is a muscle cell. This is an intestinal cell with villi projecting out of it. And this is the bone marrow, the megakaryocyte in the bone marrow, from where blood cells arise. Now, in a very basic sense, if I remove the cells from this picture completely, what you essentially have is DNA, which is a double-stranded structure told as a helix. That DNA has information. This instruction manual is the same in all cells of all our bodies, right? From this DNA, a molecule known as an mRNA or a messenger RNA is made, and this is the product number 1, the first product this instruction manual makes. Then from this product, because of this product, based on what information is present in this molecule, a second product is made, which is a protein, and this protein is different in different types of cell, which is why all our cells tend to behave differently and function differently. Now that's one of the first concepts. Now, in this entire process of flow of information, if something goes wrong, you need to be able to see it, like detect it, right? So this process is called as expression of a gene. So this is a gene, this is the expression of that gene, the product that gene made. Now what does one see? How does one see if something went wrong in this entire process? You need to be able to tell, has this process occurred normally or did not occur normally. So for today's talk, I'm going to give an example of timing of events during development. And an example I picked to show the example I want to discuss is the ultrasonogram that pregnant women undergo which tells you that at 5 weeks the fetus looked like this, at 9 weeks the fetus looked like this, at 12 weeks the fetus looked like this, and so on and so forth till 40 or 39 weeks of gestation. Now anything that perturbs this time cycle will affect the progression from one stage to the next. So for instance, maternal nutrition will affect growth of the baby. So well-fed moms versus moms who face nutritional deficit, this ultrasonogram will look different. So you know that there is a problem with the progression from one phase to the other. Now in development or in general, these events are called heterochronic, which means that there is an alteration in the timing, rate or duration of a developmental process in an animal with respect to a normal condition, a control or normal condition. So these are called heterochronic genes. So when you read about 2024 Nobel, you will come across the phrase that these two scientists, Gary Ruvkun and Victor Ambros, studied heterochronic mutations. Okay, so they were not working on human embryos or Homo sapiens as a species. They were working on this model organism, which is known as C. elegans. So like human fetus, human babies, the C. elegans also has a life cycle. So you start with an egg, it grows into one larva, then it matures into a second larva, a second stage larva, then a third stage, then a fourth stage, it becomes a young adult or a juvenile, and then the full adult, which will then continue to lay eggs, and the cycle will keep repeating. Okay. So in this frame, you can see worms of different sizes. So this is smaller than this one. There's a few that are crawling around, like this one is tinier than the other one. So these are all larvae of different ages or stages in the progression of the life cycle of this worm. These worms also happen to be what are known as hermaphrodites. We have our species has two different sexes, male and female, not genders, biological sex, male and female. And hermaphrodite, the gonads, the ovary and the testes exist in the same animal. So these animals can self-fertilize. So a single worm, the sperm and egg can fuse and give rise to the zygote that Balaji mentioned in my introduction. Now here I'm showing you fertilization. So this sperm, so this is in the hermaphrodite. What is shaded in blue is the testes. What entered the testes is the egg. So that's the egg. You will see it squeeze into the testes. As it squeezed through, it got fertilized. And this is a fertilized egg which is dividing inside the body of the worm. So these worms are optically transparent. So you can do microscopy of this type to know what is going on in an adult body, in the adult worm's body. The second movie is, and I've pointed to an arrow, which is a tissue of interest. That tissue won the two scientists a Nobel. So from this tissue, the egg squeezes out, right? So you can see two eggs being ejected out of the body of the worm post this fertilization event. There are two different worms. Here you can see cells that are already dividing, and then in a few minutes, this thing is going to protrude and emerge out. So this is the egg laying behavior in a C. elegans larva, C. elegans worm. Now this tissue is the vulva of the C. elegans hermaphrodite, and this tissue comes from a group of 5 or 6 cells that are set aside. So zygote is that one cell structure which emerges when the sperm and egg get fertilized. The egg is fertilized by the sperm. Now that one cell will divide over and over again to give rise to all the cells in the worm's body or our body, right? Now this particular group of cells are called vulval precursor cells or VPCs, and they are held in a condition where they are not allowed to divide till the larva goes from L1 to L3 stage. So at the L1 stage, these cells, these 6-7 cells are held arrested. They do not divide. And that stall in division is important because it allows other cells to divide. Okay. So if you mess with the timing of division, you either have too many of one cell type and too little of the other cell type, which is not good for the optimal cell composition of this worm. So the trick here is, there is a protein called Lin14, which when present, it does not allow these precursor cells, these vulval precursor cells to divide. Okay. Now when this protein is gone, now the cells begin division. So you need to eliminate this protein for the cells to enter the cell division program. And this elimination of Lin14 protein occurs during this transition from L1 larva to L3 larva. So you can just see it on a plate that this larva are transitioning. Now both Ambros and Ruvkun, they worked on 2 genes. When they were postdoctoral fellows, they worked on 2 genes called Lin14 and Lin4. So what I have shown on top here is a picture from one of their papers. So this is what a wild type or a normal worm would look like, okay? And we work on, my lab included, work on conditions known as mutations. So this is a mutation where the Lin4 gene, one of them is mutated. Its function is lost. This is the other mutation, the Lin14 gene. When its function is lost, this is what the worms look like, okay? And remember, Lin14 protein needs to disappear for the worms to continue to undergo normal development, okay? So when you do not have Lin14 itself, the protein that should actually be gone, what happens is that these precursor cells now begin to divide. Remember, the protein needs to be absent for cells to divide. So when the function is lost, these cells divide precociously. All subsequent cell divisions go wrong. These worms have too little of some cells, too many of some other cell types, and overall the worm is smaller, okay? On the other hand, if you have this other gene, Lin4, which is a completely different gene, if you lose that gene's function, the VPCs did not divide, which means that this Lin14 protein was not removed, right? Lin14 must go for this program to get executed. Again, all cell divisions went wrong. The vulva did not form because Lin14 protein continued to be present in these cells. The worm body is now filled with eggs. So all these little tiny circles you see are eggs, which this worm is unable to put out or extrude out into the environment. But what these two phenotypes told Ambros and Ruvkun is that in the Lin4 mutant, there is more Lin14 protein present, right? Otherwise, they would have transitioned. And the function of this second gene, Lin4, is to actually ensure elimination of Lin14, okay? There's a negative regulation. You need Lin4 present so that it can inhibit Lin14, remove it from the system. And when Lin14 is gone from the system, cells will begin to divide and everything is normal. That's the thought process that they had. This was their hypothesis. Then subsequently, so this is coming back to the scheme that I already showed. You have Lin14 DNA, it makes Lin14 mRNA, it makes Lin14 protein. Yeah. Somehow you need this protein gone so that the precursor cells can actually divide. Now, the prevailing view at that time was that the Lin4 protein, the one where the worm was filled with eggs which it could not deposit out, this protein must bind to the DNA segment of Lin14. So, the Lin4 protein binds to the DNA of Lin14, prevents this DNA from making its mRNA, and therefore this protein is missing, is gone from the system. Okay, that was a prevailing view. And both of them, when they set up their independent labs, they worked extensively to figure out what is Lin14 doing, what is Lin4 doing, and to figure out this connection. Okay, despite several years of work, they were unable to prove that actually Lin4 is a protein. They could not prove it. So from their papers, so these are old papers which are in black and white, so the figures are very informative, but they don't look very pretty. It's just a series of bands running on gels. So I decided not to show the gels, but I'm going to show you the data. Okay, so how does one decide whether a gene is a protein or not? Now, what they did was, what we also do in our lab is, in a mutant, you can add back the normal thing, the normal gene. Okay, so in the background, this is what genetic rescue is. So all these CRISPR-derived therapeutic strategies and all, this is what it's trying to do. You have a mutation in a gene, you provide back the good copy so that the mutation effect is missing, is gone completely or erased. So in this Lin4 mutant, you can give back Lin4 RNA, normal RNA, and sure enough, the Lin4 normal copy restores this mutant into a wild type like condition. Okay, everything is good. Okay. Now, how does one actually test it? So in an experiment, you need controls. So one way to prove that actually Lin4 codes for a protein is to do what is known as a frame shift. So I'll introduce frame shift first. I'll introduce frame shift. So you've given back Lin4, right? And remember, DNA is this coded piece of information. That code is actually read in blocks of 3s. So 3 will encode one amino acid or one part of a protein. Now this is a normal, this is not Lin4, it is some random example I've taken from a website. So this is the codon. So they are in red in threes. In threes, they will make certain amino acids. And at this juncture, instead of CAA, there is a C that got inserted. A C got added. When that C got added, everything beyond that C shifted by one letter. And because this code is a triplet codon, any addition or deletion in odd numbers, 1, 3, 5, 7, it shifts the frame completely. If you delete it in a particular order, then the frame is restored, right? So here in this example, because this single nucleotide got added, everything after that addition changed. So the net product, which should be a protein, it can either be a truncated protein because a stop codon got introduced, or it can be an abnormal protein which does not function. Okay, but essentially this is what a frame shift does. So to prove that Lin4 gene indeed codes for a protein, what they did is they did a frame shift. They introduced frame shifts into the Lin4 gene. Their assumption was that once the frame shifted, their rescue will not work because now Lin4 cannot make its normal protein, right? That was their thought process. But amazingly enough, even if you add back a frame shifted Lin4 gene, you still got rescue. Now this is impossible if the Lin4 gene product made a protein, because you need normal protein to function. If this Lin4 gene indeed coded for a protein, this experiment would not have worked. So I'm condensing several years of their work to a couple of data slides. Now that is a Lin4 protein. Normally, what they essentially discovered was that this Lin4 gene actually codes not for a protein, but for a small RNA called a microRNA or a mini RNA. Okay. So there are two genes. Lin4 DNA makes Lin4 RNA, makes Lin4 protein. This Lin4 protein, as long as it is there, those precursors will not divide. You need to eliminate Lin4 protein for the cells to continue to divide. There's a second gene, which is Lin4 DNA. It will make Lin4 RNA. This RNA is not coding for a protein. In fact, this RNA's only job is to make these tiny pieces of additional other RNAs. Now, how does this small RNA actually eliminate this protein? Eventually, you have to remove Lin4 protein via Lin14 protein via Lin4, right? The only way that it can do is this has to somehow interact with the Lin14 itself, okay? And that's what they showed. Gary Ruvkun's lab showed that, and Ambros's lab showed that this tiny part of this Lin4 microRNA actually does bind to Lin14 mRNA and prevents Lin14 mRNA from making its own protein. Okay, that's how in the system Lin4 protein is eliminated completely. And once Lin4 protein is gone, cells will now begin to divide and make normal structures. 14. Lin14 protein is eliminated by Lin4 microRNA binding to Lin14 mRNA. Now, where is this binding? So this interaction between this small portion of the Lin4 microRNA and Lin14 mRNA happens to be at the tail end of the Lin14 mRNA. Okay, so this is called the 3' untranslated region of an mRNA. So mRNA, if you stretch it out and lay it end to end, it will have some anatomy. It has a 5' end, a middle, and a 3' end. So that 3' end is called as an untranslated UTR or untranslated 3' UTR. In that UTR, this Lin4 gene ka some basis find complementarity. So that is what is written up in this box here. This data was published in back-to-back Cell papers from both of their labs in 1993. Now, I'm going to now come out from the data part of it. So, in a sense, in Lin4 mutants, what had happened was the Lin14 mRNA. Remember, Lin4 itself did not get made because it's now mutated. Now, in this mutation, in this mutant, the Lin14 mRNA continued to make protein. And that's the reason in the Lin4 mutant condition, you do not get vulval precursor cells to divide in a timely manner. The vulva does not form and the worm body is filled with eggs. In the converse condition, when Lin14 itself is lost, cells began to divide precociously, earlier than they should, and it came at the compromise of some other cells which should have also divided, which resulted in a deficit in cell fates in this embryo, resulting in overall the worms getting smaller. Okay. Now I'm going to come to the last couple of slides. Like, why is this relevant? Like, why are we here? Why are we talking about this? What is the big deal of this entire discovery? Because it seems like, yeah, tiny base, small RNA binding to other RNA. So what? The so what of this is first of all, it's a very, very, very tiny sequence that this thing recognizes. Amongst the billions of bases that are present in genome, for a microRNA to effectively target an mRNA and stop it from performing its activity, only 8 bases are required. So these 8 bases are known as the seed sequence. Of course, there are accessory sequences adjacent to it which ensures that the microRNA will only target its actual target and not anywhere else, right? But there is an entire machinery in place where in this case, for instance, the Lin4 gene is made into an mRNA. Then it is processed inside the nucleus by a couple of enzymes called DROSHA and DGCR8. Then from there, this product comes out of the nucleus where it is cleaved by another interesting protein called dicer, because its job is to like dice this RNA into smaller bits. And one of these strands is actually going to go bind to the target mRNA. As long as it has an 8-base similarity, it will effectively target that mRNA and result in downregulation of whatever that RNA's function is supposed to be. So this is a ubiquitous process. It occurs in all systems that have been studied to date. And we now know that this is a 2021 or 2020 review. We now know that in all metazoans, microRNA exists. In fact, there has been a addition of new microRNAs at critical points during evolution. So after Lin14 and after Lin4, which was the first microRNA to be identified, the next microRNA to be identified was Let7. So from 1993 till around 2000, nobody actually paid much attention to these papers. People essentially ignored this work because, I mean, who cares if there are 8 a tiny base pair, 20 base RNA in a worm that is not able to lay eggs, right? Why should anybody care about that? But the people began to sit up and take notice when this Let7 microRNA was discovered, also in C. elegans, but was found to be conserved across evolution. Now, additionally, at this junction point, so these are like family trees. So exactly like you will have family trees that you can trace back to your ancestors. Evolutionarily, we have family trees which we can trace back to our evolutionary ancestors. This is one such tree. So 20 microRNAs have been added here. 56 other microRNAs have been added in this vertebrate lineage. 40 more microRNAs got added in the mammalian lineage, and a few more got added in the primate. So as complexity evolved during evolution, the mRNA repertoire kept getting adding more and more microRNAs as time went by. As of 2022, we know of 159 clusters of microRNAs in our genome. Okay, so this is chromosome-wise distribution of microRNAs. The most extensive cluster is on the human X chromosome, and we are still trying to figure out it's only a 20-year discovery, right? People are still trying to figure out what it actually means. But we know wherever you look now, microRNAs are extremely prevalent. So this is a TCGA, it's a database of cancer cell types. The green indicates microRNAs that are downregulated. I know you cannot read the text, but what I want to show is that there is a variety of cancers where specific microRNAs go up or specific microRNAs go down. And these are now being used as biomarkers from liquid biopsies in patients. And of course, they are targets for therapies as well. There are several neurodegenerative conditions where microRNAs are known to play a role in disease progression as well as development. So for instance, there's a whole host of microRNAs that play a role in Alzheimer's disease, starting from transport within neurons to formation of these neurofibrils, which are these entanglements that occur in neurons. And they are a hallmark of neurodegenerative disease as well, which because they're tiny, they can pass through things easily. So they are found in serum from these patients, and diagnostics are being set up to see them as biomarkers of disease progression. Now, there are also therapies that are used to targeting microRNA. So this is again a variety of genetic and lifestyle diseases. Some of these therapeutics that target microRNAs are actually in clinical trials and will be available soon. I think 1 or 2 are already available for use. Now, why was this discovery? So I said a lot of things, right? But in essence, why was this discovery such a big deal? It did 2 important things. One was it completely turned on its head the notion that gene regulation is driven by proteins. It opened up a completely new way of looking at how information can be regulated and flow of information can be changed. The second is it led to a series of discoveries that these tiny RNAs are not new innovations that came about, at least in us or in worms, but they exist from the time of evolution, like from the time of metazoan evolution itself. And I'm a basic developmental biologist, so I want everybody to remind you also that we are here today simply because two scientists wanted to figure out why some worms could not lay eggs. That's the bottom line. I'm happy to take questions.